times into the newest part. So therefore, the title also is so promising and so challenging. But let's see. When we start with tractography here, so this is the brain. We know that. And we have the MR scan,
Tim,
and we
you're
have...
not. Sorry, Tim, can you share your
yeah,
screen?
I'm not sharing screen.
No.
Okay.
Oh, yes, you are.
You
I can
are?
see
I
it.
can see him. Yeah, you probably
Oh,
got the
sorry.
wrong window.
Oh, probably. Okay, perfect. Sorry. Great. Never mind.
Okay, I continue. Thank you. So this is the brain and the MR scan, and in there we have the gray matter and white matter, and we know we also have these connections here. So we bind them, and we are pretty interested to map out these connections here in the living brain. One of these connections here, of course, we know that this is the neurons. So we have the axons, it's the myelinated axons that are making these connections between the gray matter regions. So if you take and cut through it, then we have this electron microscopy. We can do the staining, and then we see these cross-sectional areas here. And from these cross-sectional areas, we can count like the axon diameters. And we get this kind of distributions. So why is it that I'm so interested to talk about this diameter distribution when we are going to talk about tractography? And that's actually because that the white matter here, they are the one that are responsible for our pathways here, are responsible for the communication flow in the brain. So we have some tracks here going up here and make the orders to move our feet in a necessary situation, like escaping here. So we need to have different kind of modulations. And with this kind of modulations, it is actually the axons that make this conduction timing mapping in the brain that secure that the timing or this functional regions come in the different parts of the brain in the right timing. And how does that work? So if we have the normal pathways here, we have the signals conducting along the axons. Here we know that the bigger axons and the more myelinated, then the faster conduction velocity we'll have. And we can be in cases where we have a disruption here, then that will affect our conduction velocity. Or we can have a completely pathology where we have a neurodegeneration here. And then with time, the axons, they will die and disappear. So that connection will disappear. So how do we actually estimate conduction velocity? So that we can do in a very simple way. So we have the diameter, and we just need to have the thickness of the myelin, and from that we can calculate the popular matrix here, the G ratio here. And then we can calculate the latency along the axons, how fast the signals are transmitted along the tract. That's dependent on the length of the tract. So what I'm going to talk about here is not about the length of the tract, but we are going to talk about the tractography, how we can actually map out these connections in the brain. And we don't want just to have the rough connections in the brain, we want to see all connections. There are different methods here that allow us to look into the microstructure and the connectivity of the brain. So we have the features in here. So that is going from the macro down to the nano. So up here we have the dendrites, and we can go down to the interaxonal features here in the nanoscale. Out here we have the image resolutions. So we have the low resolution in millimeters. That is typical the MRI part. And then we could go and see directly the features. That is the light microscopy, electron microscopy, and synchrotron imaging. So we actually can see directly. But actually what is unique about MRI, and specifically diffusion MRI, that we are talking about here, is that we can see things at a lower level that we actually, even that the resolution is low. So we can make this categorization of what we can see of features here. So in this, with MRI, we can see this directly, these major pathways here we can see. And if we zoom into these, then we could go to the smaller features here. That is the axons and the organization. Then we can look at the light microscopy, electron microscopy, and microstructure imaging in this regime here. So if all these things here, of course, related to each other. We have all these things we see here is related to the pathway. So how do we actually map this pathway? And that is a thing I have not looked at for many years. So I went back to my old slides. Actually, this was a presentation at the ISMM educational talk, I think in 2009. And this is actually how the basic principles are of tractography. So we have scanning. A subject we put into the scanner. We acquire some scanning. We have a fiber reconstruction of multiple fibers, and then we could do kind of tracking here. So we have two different ways, basically. This is the deterministic, where we start in here, and we just take small steps here, and then we draw the streamlines. And then we have the other one, probabilistic, that it can be shown in different ways, but we have a kind of uncertainty in each step here, and then like sampling out the pathways. So the output of the probabilistic, this is at that time by Alexander and Parker, they show this is the Pico maps. It's actually a streamline count. So we have the seeding region up here, and then we have, you can see the count of how the streamlines are projecting from this seed region. Deterministic, we just see the streamlines in a nice ways here. Today, we basically do the same thing, I would say. We are mixing these different features together. So we have a deterministic, which is mixed with a probabilistic, but that is a different talk. When we want to validate this kind of things here, then we look into these integrate tracers. This is early work of Luguet here, where they have two tracers in a monkey brain. They cut it in fine slices, and then they reconstructed it in 3D, and then they could do a validation with the MR scans that were collected. One of the challenges with doing this is that you do two traces in the same brain, so you get a mix of these. Many tracers projections share the same pathways, so it can be challenging to separate. And then we have a more extensive work here. This is the Allen mouse connectome atlas here, where they have make single injections here in the mouse brain. They have more than 1,000 injections here collected, and you could go in here, and then you can actually see on a standard brain how the tracer are projected from each of these injections that they have made, and that is something continuously that they're expanding. We also did validation at that time. That was with an MR scanner with a manganese MR sensitive tracer, and also an invasive tracer here. So this is in the pig brain. We did the injection site here, and you can see the trajectory. At that time, we also need to validate diffusion MRI. And this small pig here, that was a small mini pig, so it was very small. It was at about this side here. And we put it into the human scanner, and these are some years ago, and we collected after 20 minutes the diffusion MRI, and this was the result we saw. And many of you would look at this and say, okay, this is actually a coronal slide of the pig brain. It does not really look like something we can use for validation. And we also found out that it didn't work. So we never published this. So you are among the first seeing our failure here at the first time. So we decided another solution, and that was going to ex vivo MRI. So we actually set up this ex vivo diffusion MRI, set up to allow this validation. And this is just an example of how it looks like in the same pig brain here when in ex vivo. So we took this setup here, and then we have our tracers, and we did 3 different injection sites here. So we have 3 brains of pig brains, and we have overlapped all 3 different tracers into this brain here. And we see the red one that is crossing from to the other side. That's the red one. All of those tracers projections seem to work with tractography. And then we have the blue one here is going into the middle dorsal nuclei of thalamus, and you can see it was like circling around here. Sometimes it ended actually terminated correct in 2 cases here. If you take another tracer in prefrontal cortex, we can see then this again the same tract here should end in medial dorsal of thalamus. It was just circulating around, which was really not the right connections. We also saw that the yellow one here should end in substanta nigra. It's a nuclei down here, but it did not end there. It just skirted around and then continued until the tract was like fading out. So what was actually happening here? So what we found out is that this tracking here will not terminate in these small nuclei. And they will simply just skirt around. And the reason is that we have a nuclei structure here, and then we have a dense pathways around here with tracking. And if we now place our grid on top of this, there will be lots of parts of volume. And these few tracks that are actually entering this nuclei here, they will have low weighting, so the path will just track around. So the other observation we saw is that the tracks were just continuing. So they're fading out, so that the number of streamlines surviving this long cycle is just decreasing that. And that is what we call path length dependency. If we look at this path length dependency, that is exactly also what we see in these first tracks here from Parker and Alexander in 2005. It's the seed region, and then you see this decrease of the count rate with the distance. It was exactly the same thing we saw in the peak brain. So we have the seed region here, and then it decreases with the distance. Then there was a time where Fernando Clemente introduced this tract density imaging. So the idea was that you have this diffusion MRI, and you can do DTI. Then normally when you do tracking, then you actually do the tracking at the higher resolution than the actual resolution. And the idea was here that you stayed in this higher resolution when you've been doing the tracking. And then you do a whole brain tracking here, and you can get these nice features out, apparently in a higher resolution, and see some more contrast between fiber tracks, which is not completely wrong. Then we have, if they also, what they also demonstrated here, which is interesting, is that if they take this tract density here and just make small ROIs here and saw the projection of the streamline from these different regions here, then you actually can see the follow individual tracks. So you can imagine if we now take this streamline here and just take that and take these streams, and then we just follow this one here as a seed and then expand it, then this will continue like segment out the tracks. Actually, this is the idea of the IST method here. So this is iterative confidence enhancement of tractography. It's a seed region growing approach based on this principle here. So you have normal tractography. This is our pig brain here, and we have a threshold here. Then how the tracks appear when you set a threshold very much depend on the threshold in here. But when you do this tractography with this region growing, then it will be much more robust to the selection of the threshold here. So you can do some analysis here. So this is the distance from the injection site. So this is this projection down here. And if we start here, then we have a high count rate of the yellow. That is the normal pico, or the streamline count. And we get to a complex region, and it will decrease very significantly here, be a very low level. If we do this region growing, where the sigmoid is increasing every iteration, then we can actually see we stay with a high count rate here all along the tract. So we get less dependent on this kind of threshold than that is set. It has also been suggested to use linear distance correction algorithms, but it doesn't work. So this path length dependency can maybe be fixed with this kind of IST approaches. But we'll look at that later on. So then we took the pig brain here and looked at many different algorithms that were coming out, software tools at that time, and we compare them both deterministic with the CSD and diffusion tensor. And the approach is that, or the result was that there was not really one tractography that was outperforming the others. And that actually also is the same result we saw in the diffusion study group tractography challenge that was initiated, I think, in 2005, not 15. Here we make a synthetic image of a human brain and generated all the tracks here, so we know exactly what type of tracks we were expecting to see. And many groups chipped in here and applied the techniques, and we learned from this and here that there are no methods that are really outperforming the others. What we saw is that the general finding there's a false positive and false negative pathways, but that is like what we expected. One thing that was like highlighted is this: you have some regions in the brain where we have this bottleneck regions where there's a lot of pathways that are going in the very narrow regions here. And and one we can actually discuss is this a structural resolution limit. That means that the resolution that we're applying is too low actually to resolve the tracks passing through it. So we can do this kind of checking here. So this is a clinker dissection by Silvio Sarobi we did in the monkey brain in this region where we absolutely can see this narrow region here of tracks where many tracks are passing through. But you can actually track them if you do a great work. And this was work by Laurent Petit, who is very good at getting out the tracks here. So it is possible, but resolution might matters. Now, when you have this kind of false positive, that you have a speculation is it because of scanning parameters? Then we did a study here, and at that time, Marco and his colleagues was coming out on a nice rich tracer connectivity matrix here in monkeys. And they also applied it they related this tracers here to an atlas. And that one we can use for validating tractography. So we collected a brain at different resolutions and different kind of orientations and different p-values. And then we did this kind of structural connectivity analysis. So what we were looking at is how different regions was connected, the number of streamlines, and how it matched with the number of labeled neurons in the different regions. And here, different colors is different p-values, and here is the different number of directions. And the entropy here is how well, how good match is between the labeling and the tracking. And then we have the different resolutions. And what we see from this one here is that at least you need to have a sufficient high resolution to have a structural connectivity analysis. What we also see is that when we lower the resolution, the entropy actually improves. The structural connectivity gets more like the tracers. And what was more interesting is that when we look at the some of the best performing here, then actually the very low resolution here performed even better than going to high resolution. So of course there's a signal to noise issue here, but that also will show the same result. So what is going on here? So what this suggests is that our larger pathways might dominate this kind of structural connectivity analysis. So they are not so much dependent on the resolution. Actually, the larger one, they will have a lower path length dependency because the number of voxels get smaller to trace through the tracks. So that could be an explanation why the lower resolution actually works in this case here. But what does actually resolution mean? So this is a very nice study here by Kurt Schilling. So he looked into this resolution. How far should we go for crossing fibers? So this is passed in a monkey brain where he did structural tensor analysis with multi-fibers. So he applied this and then looked into different resolution. Going to higher resolution, what happens? Do we still have crossing fibers? And what is super interesting here is that this regime here, this is where we are in the MR scanners. When we go lower and lower and lower, all the way down to 32 micrometers, what they tested here, the crossing just get more and more severe. So that is interesting. So how can that be? So then we looked into what level should we go to before crossing fibers become single fibers. And this experiment we did with MRI. So we have a half a micrometer here, an isotropic DAISY, a synchrotron facility where we go to half a micrometer, and then another synchrotron facility, ESIF in Grenoble in France, where we go to 100 nanometers of the same sample. So what we see here is this with a diffusion tensor that in corpus callosum is straight fibers. And this part is where we did the sample. And this sample is a half micrometer resolution in straight fibers. And what we see is that we have this kind of laminar structure. So even that is aligned fibers, then we still have a kind of 35 degrees of laminars that have an inclination angle here. And this thickness here, they are about 40 to 45 micrometers. If we did the same in the mouse brain, we did not see this kind of laminars like what we see in the monkey brain. So we cannot say that they have an inclination angle, and the thickness we don't know. But what is interesting is that Su et al., they actually explored organization in corpus callosum here with two neuronal tracers, so you have different colors. So you can actually see in the mouse brain, they are organized here, but they are running in parallel. So that is why we don't see this laminar here with structured tensor, even that we have this very high resolution, simply because these laminars are in parallel. We can go into crossing fibers. So this is how it looks like here. This is in the MR scan. We go into syndrome Simowell, where we know we have 3 crossing fibers. And if we take this crossing fiber here and use, here is the CSD, we get this FOD within the voxel. If we take this sample here, this is the sample we did synchrotron imaging. We applied in this super high resolution structured tensor analysis. Then we get this result. So the color coding here describes the direction, the principal direction of each small tensor in this regime. And what we see here, what we have highlighted there, is that we actually have 3 crossing fibers. So, but they are organized in laminars. So this you can see is like there's one laminar here, there's another one here, blue, and then we have greens going in another direction here. So we have a very complex structure here, but it's very simply organized in laminars when they're also crossing. So if you take this whole volume here, which actually covers an MR voxel here, then you get an FOD in this volume here from all this direction here that very much looks like the FOD at the voxel level with an MR scanner at a much lower resolution. What we learn here is that when these laminars were intersection, it is like on a grid-like structure. So it's around 90 degrees. And the thickness here is ranging between this 5 to 10 micrometers and even thicker. So what we can see here, that this FOD, they actually scale across these kind of resolutions. But what we are missing here is the subvoxel spatial and this laminar organization, which is important for tractography. So how is this organization actually been reported throughout time? Is this laminar structure and is it this grid-like structure something that we should actually take into account? And I think that's the case. So actually many years ago, Van Medeen published this work here in Science. And he showed that these different major pathways here, when they cross on a grid-like structure here. What we then can add to here is that it's not only a grid-like. We also have pathways that are very organized, but they follow here in more parallel structures, like what we have shown here with tractography here in these fibers here. So if you zoom in this tractography here, then it's actually also been described in anatomical studies that when you have this is this deep white matter here, syndrome Simwell, we have axons projecting in, and it's been illustrated they have a very sharp bending here, almost like a on the grid-like scale when it turns off, what we have seen here. So it looks like there are this kind of grid-likes, either parallel or grid-like. And this also supports our finding here, what we have found in the monkey. So even that they have parallel fibers in corpus callosum, they can actually still have this laminar organization, but have a different angles here, or the crossing here, which this region is central symbol, which is similar to this one here. So how is this grid-like? Is it just noise in the literature? Well, I don't think so. And this is what coming out. So this is actually called fasciculation or defasciculation. It's also called a zippering or unzippering effect. And this is something which is happening when the tracts are formed at the earliest basis in brain development. So this is Smith and Elf. They published this video here, and I will show. This is in a small population of neurons that are sending out axons here. Let me see if I can get it started. Here. So what you see here is all the actions that are formed. These are formed. It looks like a bit chaotic in the beginning. But then what is happening is that they have like a zippering effect. So you can see they start to collapse into fibers. So they try to align in a way this one here. So you have a leading neuron that's sending out, and then fibers that follow the same direction will try to lap into one fiber. And then in the end, if they need to divert out when they got to production, then it actually can split out here. That is the unzippering effect. So this is only happening before myelin is coming out. The effect disappears when the axons are myelinated. So this is another example where they show more this effect here. This is the unzippering effect. So they follow and then they zip out. When they cross on a grid. And that is because that is the time where this chemical physical interaction that is determining the forming of the fibers, that they have the best where they are somehow more independent of each other. Then you get this very grid-like crossings. So it might be that this is a simple rule of anatomy. So one thing is here, what we see here of this organization. Then we need to capture this with an imaging technique. Because of this structural resolution limit, we need to go down and be able to capture these individual things to do tractography to work properly. There exist different ways that are label-free. So only talk about the label-free orientations here. And this is our good friend, our diffusion MRI with DTI. And this is Johnson here that recently showed that in the mouse brain, they managed to collect this isotropic 15 micrometer of resolution of a whole mouse brain with their MRI set up. There are others that have also shown in a direction, or one thing to say, maybe this is also the limit, because when you go to this resolution, you will start reaching the diffusion length of the molecules what you set up, but that's another thing, it's just to be considered. You can also go to high resolution with this polarized light imaging that has been presented by Marcus Axer some years ago, where you do the birefringence of the myelinated axons. You have light penetrating through the slice, so you have slices, and then you actually can estimate the fiber directions from the organization of myelinated axons. It has a limitation not to be able to detect crossing fibers. And that is a newer approach here by Myron Menzel here that proposed the scattering light imaging measurement. So it's still a slice where you light through it, but it's using a scattering light principle. And that allows you to actually not only model single fiber, but also crossing fibers. And so this is what they presented here. One thing is about these techniques here, they're not really 3D because they have some dark spots on the fiber orientation profile that we just need to be aware of. And also it's slices. So when reconstructed into a 3D, it can be a very challenging structured procedure to do this. But I think there are some approaches to make this happen. Then there's another one that overcome a bit this problem with aligning the structures afterward. And this is this PSOCT approach by Lou et al. That recently published this one here. So what they're doing is that they're scanning the surface of the brain, and then they have a laser going down, and they actually integrate the fiber orientation in a slice-like way down in about 150 micrometer resolution. And they have a higher in-plane resolution, about 10 micrometers. It is based on the same birefringence principle like polarized light imaging here. So it is still single fibers image, but it's a real 3D fiber orientation that they can extract here by measuring in different angles here. This slices here is taken from a block phase. So you image and then you cut away the slice. So you can easily reconstruct the 3D of the fibers here, and this show these results here. But if we want to go to this laminar organization of the fibers, if we need to go to imaging technology that can go to even lower resolutions here to handle these laminars, which are around 5-10 micrometers. And this is a new technique we have been working on here. So it's really about imaging the invisible. So the story starts here. This is a movie, Griffin, The Invisible Man. It's a scientist actually, and he developed a new drink called monocaine. And when he was drinking this, he actually became invisible. And this was a very nice science fiction movie here in 1933. But actually, some years before that, there was Wells that wrote this book here about the Invisible Man from 1897. And then you can all think about, what is the point here? This is a bit spacey. But actually, this is not completely off. There was already in 1911 here, there was an anatomist, Weiner Spatelholz. And he was trying out different chemicals, and he found a chemical where it actually managed to clear hand. Or this was the bones. So when he light through the bone, then he could actually see the structures inside. And this is the technique what is has today advanced a lot on clearing tissue. But the thing is here, when we have this clear tissue, so how do we actually image these things here? And just for illustration, so we have removed the head here, and we have the invisible things we want to image. We take a camera on top, and then we have a laser sheet. We go in here, and we take an image with camera. This is one slice. Then we move the laser sheet. We take a new slice, and then we move the laser sheet, and then we get another one. This is the principles of light sheet imaging. So light sheet imaging here uses these chemicals that clear the tissue. So this is a normal brain. And then you can use different chemicals, and there's a lot of different protocols here where you can clear the brains, and it gets transparent here. Then you have this light sheet microscope where you can light through it, and then you can move the light sheet, and then you can image in 3D of this intact brain, these structures here. So this is labeled imaging. So this is for the neurons and blood vessels, and you can combine them in the same acquisition at different wavelengths. And you can also do this kind of clearing of human brains. So this is an example here. Human brains is a bit more challenging because of the myelin, how it is to get it cleared. But this is actually something which is really established things that has been worked. But for some years ago, we discovered that you can take this clear brain here, and then if you set the light sheet into a scattering microscopy mode, then if you place it and you rotate the brain in this sheet here, then you will actually see that you have intensity changes with the rotation of the brain. And when you reconstruct this different rotations, then in each voxel you will get like this kind of fiber profiles, like what we see in diffusion MRI. So this is in white matter region, and this is a corpus callosum, which have another orientation, right-left. We have a white matter bundle here, also have another orientations. If you look in cortex, you will also get anisotropy, but it's a low anisotropy, like in diffusion MRI, but you still have some kind of orientations, and it varies between the different regions. So you can actually visualize this contrast of the directions, dependence of this contrast in the brain. So each measurement, each direction here, we can then color code from 0 up to 180 when you rotate in the brain. So you can just see the different parts of the tracks here, dependent on the orientation, get a different color code here, dependent on the orientation related to the light sheet. This is not DTI, it is raw data that we are showing. Okay, so we can actually collect this. This is for isotropic 4 micrometer resolution in the intact brain. Then we take a diffusion MRI acquired in 125 micrometers, and then we have 4 micrometer resolution here. And this is the result we get out in the same brain. So this is a coronal slide of a mouse brain, and this is a tensor model fitted to this light sheet scattering microscopy. And you get fiber directions here in 3D. And you can see the fine details that you get in this color-coded FA maps here compared to the DTI. And if you zoom into different regions, so we have here the strial fibers, you can clearly see they are coming out as bundles. In the MRI, they are removed because you can see them because of partial volume effect. And similar here, you see fine details of bundle of fibers that you cannot resolve here with diffusion MRI. So it really goes to high resolution. So this is 4 micrometer isotropic. And basically there's no limitation. You can go to higher resolution here if you want to. But this is what we could acquire with a field of view without really need of stacking things together. So we also tried to explore how this contract changes with my demyelination. So we have a mouse brain where we introduced a demyelination, and this is where you have the demyelination sitting here in corpus callosum. So this is a maximum intensity projection. So we actually have an impact of the signal on the scattering light. Then we can see the directions. The directions, you still see directions here. And if you calculate the fractional anisotropy, you can calculate the fractional anisotropy in this map here, like what you do in DTI. You can see it's lowered here. We can also see when we combine the same brain with the myelin basic protein here that there are no myelin here. It is removed here in this lesion region here. And then we have the region here where we just show that we have a lot of cells coming in because it's like inflammatory response. We have this lesion situation, and that can explain this lower fractional anisotropy. We can also do it in human tissue. So we have a human tissue, a small chunk here. And this small part here, we cleared it, and this is the maximum protection of this 4 micrometer resolution of the fibers in corpus callosum, and you can actually see the individual fibers and also crossing here. So in this small region here, if you apply diffusion MRI, so this is diffusion MRI, this is in 125 micrometer resolution, and this here is this sample. So these is a big voxels compared to what we can go into 4 micrometer resolution, where we can actually resolve the individual fibers here. So this one here is the same region covering here. And if we zoom into this voxel here, we still see crossing fibers. And you say, okay, there's something wrong here. But it's actually right. You can zoom in, and we have a synchrone imaging showing that you actually have crossing fibers in this region here. So that means that we need, even at this resolution here, to increase this kind of crossing fiber. So this is Celia here. I've been working on crossing fiber models. So this is the FA map. So this is cingulum bundle with a tensor mapping, the single fiber, and this is corpus callosum. And we have this region here of low FA fibers. If you take a crossing fiber, then we have the first fiber here, and then we have the crossing fibers. What is interesting is there are actually situations where we only have single fiber. This is here, but we also have a rim and a transition here with crossing fibers. And what is interesting when we look at the crossing angle here, they are all very highly crossing, so they are on the grid level. So taking this further, we can do tractography. So we can take this crossing fiber light sheet microscopy and use a tracer here from Alan Mau's Atlas. And we can take our mouse and see if we can get from the same seed region here, if we get something similar. So this is downsampled to 50 micrometers, and this is 18 micrometers. So we don't have the full resolution of the 4 micrometers here. So what we see here is that we actually can trace something that looks like what we have in the tracers. But we also see that the higher resolution, we have this path length dependency. It's hard for the number of streamlines to survive the length here of the tracks. And when we go to even higher resolution, the challenge becomes larger. So the question is now that we can go to this structural resolution limit to see all these fine structures, but then we have a chance here about the path length dependency. So everything is good, but there will always come new challenging up. So we can summarize this. So tractography, so promising and so challenging. So I presented this structural resolution limit. So it's really about when we are talking about resolution, people talk about we change a resolution from 2 millimeters down to 1.5. But what does it really mean? So when we talk about resolution changes, we should talk about it in relation to a structure. And this is the same thing here. This is a tracer study from Sharma and Pandya. So I've just placed a grid on top of this, and you can see the size of the voxel matter what we actually can see of partial volume effects. So therefore, we have to talk about the structural resolution limit. We can see that this pathway here, they are actually structurally organized across length scales in these laminars, and they can be very small up to our major pathways. So does crossing fiber solve our problem and our challenges? Yes and no. If the resolution is too low compared to the organization of fibers at the final level, then we lose all the spatial information and it does not solve it. What we actually see here is that the crossing fibers is really something that we go towards something which is grid light or parallel light across scales. And with very low parallel light is 0 degrees. Then we can see how can we react to these higher resolutions. This is where I introduce this light sheet scattering microscopy imaging. There's no labeling and there's no limit by slices. So it intact slices that we actually can do. And it's sensitive to multi-fiber sensitivity. But the luxury here is that now you can go to something very high resolution and in 3D, but you also have the new chance about the path length dependency when tracking these kind of things, which is something I think we somehow have to, that's an issue we need to solve. So this was the summary. We just let the movie here finish. Beautiful stuff, of course. And then thanks for, this is my group, active and all the funding. And I also have to say there have been many people over the years that have been contributing to the work I've been presenting here. So thanks to all these guys. So that's it.
Thank you, Tim, for the nice run through all these beautiful studies. Okay, we have about 80 people or 70 now, so I'm sure that we can get a nice discussion going. There are some questions in the chat, but I also want to give the chance to, yep, Laurent, please go ahead.
Okay, hi everybody. Thank you very much, Tim. This was a very great talk. Very impressive. I was wondering, when you are speaking about the grid notion, do you think that we are speaking about the same type of grid when we are comparing the grid at the axonal level and the grid at the Van Weyden level, let's say?
It's a good question. I think so. I think they are related because when you're zooming down at the axonal level, what you saw, things formed up like bundles, and it still follow this basic principle that they are either very much crossing or they are very much like parallel. So, and of course, if you zoom out and zoom out, then you will make a partial volume effect of this effect. And then you will get some effect of the FOD at the voxel level at the low resolution, where it will not appear about this grid light. So that is where you start to have different angles, I think. So I think it's related to it.
I agree.
I have a curiosity. When you do the tractography, the minor curiosity, when you do the tractography on your light sheet scatter method, is the actual orientation of the fibers that you can recover that you follow, or is it some sort of structure tensor? No, it's the actual orientation. You recover an angle.
No, yeah. So it is. It is the orientation, but you also have an FOD around it. So it is like converted into a spherical like fiber orientation profile. So we are not looking only at the fiber direction. So it's not like that you have a stick that you follow. You have a FOD around it.
But do you do that? To do that, you sort of like downsample, so you use the pixels or voxels around, or no?
No. This is voxel-wise. So we did it now because it's just to testing in the lower resolution. When you go to the high resolution, you have so many problems. But it's the raw. The profile is a bit. There's something in this scattered light with this microscopic with a light sheet that is not completely aware what is it actually generating this signal here. So you can go to very high resolutions, and that is something we need to go to look into. So the profile of the fibers means something, and that is something to be explored.
Interesting. Yes, Martin.
Yes, nice talk. Thank you very much, Tim. I was wondering, we discussed that before, but I'm wondering if there's any new tracer. What tracer would you use to see in the MRI, and also that could be used for track tracing in postmortem monkeys? So is there anything that would be better? You mentioned a few, I think, but I'm just wondering if there's anything that you would suggest.
Yeah, so we have been talking about it. I don't think there exists a tracer. There exists this dye tracers in postmortem, but they are very slow to penetrate. So I think this in vivo tracers, like what Allen Institute are doing, or what we other people are doing with tracers, in vivo tracers, and then you stain it afterward. That is the way to go for.
Yeah. Okay. Thank you.
There's another thing is, of course, also you cannot inject a tracer in the human brain. That is a challenge. And these other kind of tracers might be possible, but it's not optimal.
Since I have you here, Martin, can I ask you a question? Can I ask you to comment on the on the grid-like structure?
Well, it's hard to comment, but we have this in mind. I mean, with Tim, we discussed that before, the option of injecting a tracer and do the track tracing after. But I think it's hard to decide on or to find a tracer that would be suitable for MRI and track tracing. So that's why if anyone has, suggestion that when we like to do single axon tracing, so it's not all tracer that allows us to do that. So it's kind of tricky to find the right tool to get there. But if anyone has any suggestion, I would be pleased to have it. Yeah.
I would also add something here, comment, there's also something discussed, is when you have a tracer, then you select what you want to see. Then it's hard to see this kind of organization that I'm talking about, the laminar organization, that you can only see if you zoom in and then you look at the region and you see the whole environment together. That you will not see with the tracers. So it's no guarantee with a tracer that you reveal this kind of information. So it is this kind of clinker dissections some have been showing, or this synchrotron imaging where you really can have a larger field of view and see reveal this kind of organizations where this will be clear.
Yeah, Christian.
Hey Tim, absolutely beautiful images as usual. Quick question. So in the images that you showed of the light sheet of the cleared brain of the mouse, there didn't seem to be a lot of anisotropy in the cortex. And I guess the question I have is, what did you observe, or have you looked at the cortex in more detail? Do you see directionality, and do you see these crossings as well in the cortex?
Yeah, very interesting question, Christian. Yeah, you see crossing fibers in cortex, and it follow exactly what has also been Katja's drawings, where you have this laminar organization, where you have fibers going up to the surface, and then you have the crossing fibers in the different layers. This structure is followed by that we also see in the light sheet. It follows completely. Yeah.
Laurent, did you want to add something? Laurent, you unmuted yourself. Did you want to add something? No. Okay. All right. I'm gonna read. There is a couple of questions in the chat, so I want to make sure I read them too. You mentioned the use of TDI to iteratively improve tractography. If I understand it correctly, this works by extracting additional ROI from TDI's contrast. Does it mean it can only be applied to seed-based tractography, aiming at specific bundles?
So the idea with this IST is based on the principle of this whole brain tracking. So it is a seed region. So you select a region where you want to extract. So normally, what people sometimes do, you have a seed region and then you have a target region, and then you find the tracks that combine these. What you're doing here is that you select a seed region and then you let it grow, and then you can filter afterward what you would like to see. So it's just a way to make it less dependent on this path length dependency, this count rate. So it should work in generally.
And Simona is another. Simona, if you want to unmute yourself, feel free. But
Yeah. Thank
Okay.
you for answering. Yes, it was very clear. And yeah, the other question was about you try to there was an experiment in which you showed the effect on the connectome of using different scanning parameters, but I didn't get how that related to the tracing data.
So in so the tracing data to that was like you have that's been tracing data, and they counted the number of neuron reaching the different regions from the injection site. So we have this connectivity matrix. And this was what we were comparing to. So we are seeding a region where you have an injection, and then you see how it projects out to all other regions here. And you get a distribution of streamline reaching the different regions compared to the number of cells you have detected with tracers in there. So you get actually two connectivity histograms between target region and this between the source region and the target region, and that is what we were comparing. We were not looking into how the tracks look like at all, only who joined from the start to the end.
Okay, I see. Thank you.
And if you would like to try it yourself, the data is free. So you can download everything and then you can give it a hit yourself.
Okay. Any other question, comment, curiosity? I can ask a general one, like, do we really need tractography to do what like micro CT does to make it useful? Micro CT or any other microscopy? Like, do we really need it to recover the laminar structure you see here?
So that's a good question. I think that is dependent on what it is that you want to do. So if you want, if you have a surgery situation where you just want to, if you want to see how the fibers is actually projecting around the axons, how they've been pushed, then you don't need to go to this resolution. If you want to understand how the brain, from a basic research question, all kind of connections in the brain, how it's organized, then what we do today with tractography is not possible. Then you need to go down. You don't need to, the brain is not designed for the single neuron. It is completely unrobust. So it is designed to have pathways with a distribution of axons that are going to the different things. So one neuron dying will not kill down the whole system. So therefore, you need to go down to the resolution where you have this laminas. And that is good for us because that means that we don't need to go down to the resolution, so we need to see individual axons. And that pays us for some pain going to this kind of resolution of images, because we just need to go to the laminar structures range.
Just.
Yeah. Oh yeah. Okay. Sorry. Just.
Okay. Well, if there are no more questions.
I think Christian has a question.
Oh, sorry, Christian. Go ahead.
Yeah, not so much a question, but I just want to take the opportunity to remind everyone about the IST conference in Banff, October 25th to 29th, and later this year. And that if you missed submitting an abstract at the last deadline, there's going to be a late breaking abstract window from July 22nd to August 19th for posters, and really encourage people to submit and send their abstracts in and hopefully attend, and hope to see you there.
Yes, good call. All right. So the other thing that I should say is that we're not gonna have a Tract Anat in July, but look out for an email for a Tract Anat at the end of August, most likely. The recording will be available as always on YouTube, and if you have any ideas, suggestions, or if you want to request someone to give a talk at a Tract Anat, please email me. With that, everybody enjoy your summer. We'll see you in August.
Thank you.
Bye-bye.
Bye-bye.
Bye-bye.